talk about multiphoton microscopy, mainly endogenous signals and harmonic mode of contrast. So, first a quick reminder about multiphoton microscopy. So, multiphoton microscopy is an alternative to confocal microscopy for 3D imaging. Instead of excited with one blood photon, we excite with two IR photons, and we obtain the same fluorescence. The advantage here is that we have signal only when we have a high intensity, that is at the focus, so we have intrinsic 3D resolution that is more robust upon scattering than using a pinhole. And moreover, we use near-air excitation that has a deeper penetration in tissues. So for these two reasons, we have a superior imaging depth, while we have typically the same acquisition time and the same resolution as confocal microscopy. The other advantage, that is a key advantage of multiphoton microscopy, is to have all these additional modes of contrast. So you see all these different signals that come from nonlinear optics and that give some other signals and fluorescence. So let's look at these signals. So when we classify the signal, usually we classify as one photon excitation, that is linear confocal microscopy, then two photon excitation and three photon excitation. So two photon excitation is usually excited around 800 nanometer, and there are two signals that are both scales at the square of the intensity. So two photon excited fluorescence and second harmonic generation, and they can be excited simultaneously and separated because they have not the same spectral properties. For three photon excitation, there are mainly two signals, three photon excited fluorescence and third harmonic generations that scale at the third power of the intensity, and here it's shifted a little bit to the near IR light, at least 1.2 micrometer, usually further. There is also some coherent antihistogram scattering, so I will not address this signal because it's done by Hervé Rignot in the round table. So it's maybe it's not, well, I've not corrected here. It's a round table on Thursday afternoon. No, this afternoon. So I will just focus on these two signals. So the advantage of three photon excitation is that while we have a similar resolution, typically, we have a better penetration because there is less scattering in the near IR, and also a better confinement in depth, so it's less, it's more robust upon scattering because it scales as a fourth power of the inverse actual depth. However, we need a higher excitation power because there is less signal, so it's a little bit more tricky to have no photodomage, and there is no mass market laser source as titanium sapphire. So with OPOs and OPAs, it's a little bit more complicated. So that's a usual separation, but we can also separate between incoherent process and coherent process. So fluorescence is an incoherent process, either 1, 2, or 3 photon. So it's well known. It's first 2 photon absorption and then fluorescence emission. And before fluorescence emission, there is some non-radiative intraband relaxation, so that there is a loss, there is no phase memory of the excitation. So it means that we will sum up the intensity of every chromophore in the focal volume, not the electric fields. So for this reason, the signal scales as here the fluorophore concentration. Of course, fluorescence quantum efficiency, the 2 photon absorption cross-section, and as I mentioned before, the square of the excitation intensity. So for these reasons, we have a linear dependent on fluorophore concentration. We have also isotropic emission. We have also, of course, a wide spectrum because of this relaxation, and also a nanosecond to millisecond lifetime. So it's completely different. Oh, so sorry. What are the endogenous fluorescence signals? So at the cellular level, here it's a cornea epithelium, you have mainly NADPH and flavin fluorescence that are molecules that are implied in the cellular metabolism. So you can see these molecules in the cytoplasm, so, and the nucleus are do not have these molecules. So it means that we can see the cells, and also we can measure the cellular metabolism, either by measuring the ratio of these two signals that are not excited at the same wavelengths, or by looking at the lifetimes. So we can combine this 2-photon fluorescence emission with fluorescence lifetime imaging and look at the lifetime of NADH and FAD, and then have a measure of the cellular metabolism. So you can see the workshop by my colleague Chiara Stringari at the lab. There are also some extracellular endogenous fluorophores, mainly, well, the keratin is the skin epidermis, but mainly elastin. So we can see elastic fibers or elastic laminae here in this carotid artery. So that's for the fluorescence. Now let's look at these current processes. So second harmonic generation, third harmonic generation, and cars. So for that, we have to come back to the physics of these processes. So it's really the basic of nonlinear optics. In nonlinear optics, we look at the polarization, that is the summation of all the dipoles in the focal volume. And these dipoles can be linear in the electric field, so that's the refractive index, or non-linear, and that's what we are looking at. For example, here we look at electrons between electroacceptor and electrodonor. So when we excite with a strong electric field, we have an oscillation, so the dipole, the electron oscillates, it's an electric dipole. But if we have an electroacceptor or any specific shape of the harmonic potential, of the electronic potential, it spends a little bit more time on the acceptor time. So when we look at the Fourier transform of this oscillation, it means that we have also components at other frequencies than the initial one. So it means we have second order polarizations that at 2 omega, so it's second harmonic generation. So to have second harmonic generation, we need, as schematized here, some non-centrosymmetry, while for the third order one, any molecule is able to radiate third harmonic generation. So any dielectric material. Okay, so the question is, what are these dipoles in tissues? So the main dipoles are the peptide bands. Here you see the peptide bands, that is the skeleton of the protein. You have some delocalized pi electrons between an electroacceptor and electroacceptor and electrodonor group here, so that we can have a small dipole that generates a small electric field at the second harmonic frequency. And the way to measure this is to measure the hyperpolarizability in nonlinear optics. Now, what we measure in optics, of course, is the signal at the wavelength scale, that is in the focal volume, typically 1 micrometer, 1 micrometer size, so that we have to sum all the signal for all the peptide bands in the focal volumes. And I remind you here, we do not sum the intensities, we sum the electric fields, because it's a coherent process, that's the definition of the coherent process, so that we may have interferences between these these electric fields. So if we have two peptide bonds that are aligned in anti-parallel directions, it means that we have electric fields that have opposite phases, and we have no signal. Where they have parallel directions, they have the same phases, the electric field is doubled, and as the intensity is the square of the electric field, it's multiplied by 4. So it means that we have this coherent summation that results in a signal that is proportional to the square modulus of the elementary response, and the square modulus of the number of peptide bands that are aligned in the same directions. So it means that also at this scale, for second harmonic generation, we need non-centrosymmetric and also a dense distribution of peptide bands. So practically, it's the reason why we have signal only in filamentous fibrillar collagen protein assemblies. Just to mention, for people interested in plants, the elementary signal is a sugar, and the fibers corresponds to polysaccharides. So because of this, this specific build-up of the signal, this current built-up because we have phase memory, we have also a specific radiation pattern. So I remind you that with fluorescence, we have isotropic emission. Here we have dipoles, that's our dipoles, and dipoles emit only around them, but not in here in the left and right side, not along the dipole direction. And moreover, they have phases. So it means that the radiation pattern will be very sensitive to the distribution of the nonlinear dipoles and also to the phase of the excitation beam. I will explain this a little bit further for third harmonic generation. So practically, we have more signal in the forward direction than in the backward direction because the interferences are more constructive in the forward direction because all the signals have the same phase. So it's measured using this formula. Here we have a sine squared with a quantity that is a coherence length. And the coherence length is typically 20 micrometer in tissues. While in the backward direction, there is some destructive interferences between the signals that are, if there is some dispersion of the refractive index, signals that are that appear in the, let's here and here, they have not exactly the same travel of the phase due to index dispersion. So we have very small coherence lengths. So practically there is more signal in the forward direction. So usually transdetection is better. It's not always the case. For example, for very small signals, for very small collagen fibers, for example, forward and backward direction are typically the same. But for larger assemblies, there is more signal in the forward direction. So just to mention something about two-photon light sheet, because in two-photon light sheet, we may use some second harmonic generation. But here, you see that there is no radiation along the direction of the dipole. So it means that if we have a light sheet that is this direction, and the detection, I need 3 hands. If we have the light sheet here and the detection is here, if the dipoles are aligned like this, so they are excited with vertical polarization, there will be no radiation in this direction. So no second harmonic generation. So it's only possible to detect dipoles that are in the plane of the light sheet. So it means that it's not really quantitative because it detects only, the dipoles that are aligned in the light sheet. Okay. So what are these endogenous signals for second harmonic generation? Only dense and non-centrosymmetric structures. So of course, collagen fibers. So I will focus on this signal. Also myosin filaments and microtubules. So that's the only 3 structures that have second harmonic generation in mammals tissues. And the collagen signal is the largest one, while myosin signal is, let's say, reasonable. This one is very small, so it's very difficult to detect. In plants, we have signal for starch, so it's quite easy to have signal from this one. It's quite high. Yeah, it's just potato starch, so maizena from my home. And we have also signals in cellulosis, but here the signal is quite small. So now I will focus mainly on collagen to have one example. So collagen, here you see human biopsy of fibrotic kidney, and you see some collagen here in green color, and it corresponds to the region when there is some fibrillar collagen. While here in the glomerulus, there is some accumulation of non-fibrillar matrix, but there is no signal. And the reason is due to the structure of collagen. So just to remind you about the structure of collagen. So collagen is composed of triple helices that form fibers. And these fibers may assemble in different structures. So it's a so-called multi-scale or hierarchical structure. So in skin, for example, it's quite large fibers that assemble in fibers in fascicle in a quite disordered way, while in cornea there are very thin fibers that are very well aligned around within lamellae that are superimposed along the depths of the cornea. So here what we see is all the peptide bonds are very well aligned within the triple helices and within the fibers, because all the three helices within the triple helix are aligned, have the same polarity. So it's the same polarity, and the same for all the triple helices within a fibers have the same polarity, so that there is constructive interferences. While at the fiber scale, there may be fibers oriented in one way and fibers oriented in other way, so that it's quite complicated, because we may have no signal just because we have two fibers that are not aligned, that are aligned in an anti-parallel way within the focal volume. For non-fibrillar matrix, of course, we have no signal because it's organized as a network. And it's the same for gelatin that is denatured collagen that has typically the same distribution. So it means that with second harmonic microscopy, we probe the fibrillar structure of collagen. It's completely different from immunochemical labeling, where we probe different types of collagen. Here it's all the types that form fibers. So it's very interesting, for example, to look at fibrotic tissues, because what is important is to look at the fibers, not at the sclerosis. But it's quite complicated for physicists, because the signal scales, as I've mentioned, as the square of the number of aligned peptides. That is, it's a non-linear response compared to fluorescence, where it's linear, so it's much easier. And also, there is issues with the 3D distributions when we have fibers aligned different ways. So what does it mean? It means first that's one of the reasons why the signal in the forward direction and in the backward direction are not the same, and it may give some information about the distribution of fibers within the focal volume. What it means also is that it's not possible to do deconvolution, because deconvolution is only possible when it's a linear response, and also the diameter measurements of the fibers are usually not reliable. So it's really complex, and the only way to perform deconvolution or to have more information is to measure the phase of the signal by doing interferometric second harmonic generation. That is, the second harmonic signal is then measured, we measure the interference of the second harmonic signal of the tissue with a second harmonic signal with a reference second harmonic signal. So this is, of course, highly complex, only 2D measurements. It has been performed in two groups. First, the group by François Lecarré in Canada, and the group of Dan Oron in the Weizmann Institute, who had done some deconvolution by taking into account the phase. But any other attempt to do deconvolution is not relevant. So, still it's not quantitative truly, but we can try to have, let's say, phenomenological scoring, for example, by measuring the number of pixels that have a significant signal, so above a specific threshold. And it's very sensitive. Why is it very sensitive? Is that because of the specificity of the signal for collagen, so there is no background noise in the images. And also there is no staining, so it's really reproducible, and it scales at the density squared. That is a high contrast. It's a higher contrast than usual collagen signal obtained by OCT or confocal. Still, it's not truly quantitative, except for isolated fibers. For isolated fibers, we have measured the signal as a function of the diameter and proves that it's the force power of the diameter, and we have also measured the sensitivity sensitivity threshold. This is typically 30 nanometer. And it's typically the same sensitivity threshold as in Raman's and in the CAS microscopy, that is typically 10 to the 6 bonds. Okay. Now let's switch to myosin, but very rapidly. So myosin, here we see the signal specifically from, well, it's here sarcomers, and we see it specifically for the myosin that is being proved by immunostaining. And when we look at the structure of sarcomere, so the basic unit of myofilaments, we see that we have these myosin molecules here that are like this. And so there are many peptide bonds along here, this region, so that there is some signal here. In the very center of the sarcomere, there are myosin molecules that are oriented the two different ways. So they are anti-parallel alignment, so that it's centrosymmetric, so that there is no signal here. And there is no signal here, of course, because there is no myosin. So usually there is a double striation of the signal. It's not always the case because it's, well, a coherent signal. It depends really on the distribution of the peptide bonds within the focal volume. And you can see these papers by Wed's group at Rennes, who studied all these different configuration of myosin filaments. Okay. So it means that we have not so much information with second harmonic generation about the structure within the focal volume. So what way to address this issue is to add polarimetry. So how do we do that? That's a usual multiphoton microscope with galvanometric mirrors and forward and backward detection. And we use wave plates in the back focal plane of the objectives. So the first one is a quarter wave plate. So from the optical point of view, it enables to have a perfectly linear polarization, while they are all, due to all these optical components, usually it's elliptical. Then there is a second plane that is also a quarter wave plate, and it enables to switch from linear to circular. So it's a better way to do some second harmonic generation is you use this circular polarization, not to be sensitive to any direction within the sample, so to have a flat response. We may also use it to switch from left circular to right circular and to do some circular dichroism measurements in second harmonic generation. So it enables to cancel the signal of the fibers that are within the imaging plane and to visualize specifically the fibers that are out of the imaging plane, and moreover to visualize the polarity disorder of the fibers within the focal volume. So that's another story that's a little bit complicated. I will not focus on it. I will just look at the linear polarization. Here we have a half wave plate so that we can rotate the linear polarization. Just to mention, usually there is no analysis because it's a sick sample, but if possible, we can also analyze and then we have much more information. To mention also that this experiment may be tricky because of because of distortion of the polarization. So because of the scanning issue, usually the polarization is not so perfect on the edges. So sometimes it's necessary to reduce the field of view. And even that, when we have deep, sick tissue, the birefringence of the tissue will distort the polarization. And of course, because of the strong focusing, there are also some distortion of the polarization. There is some, for example, some actual polarization. So it's possible to take into account the effect and to correct them. Another issue is that it's a slow technique because we have to record different images, but it's possible to replace this polarization control by a very fast switching using electro-optical modulator and to synchronize this switching with line scanning so that we can have much faster measurements in moving tissues. So here you see the stack of images recorded with different polarizations that are indicated by this direction here. So in every Z position, we have a stack of images. And then in every pixel, we look at the variation of the signal as a function of the incoming polarization. And then we know, using a tensorial analysis of the second harmonic signal and using hypotheses about the symmetry of the collagen fibers, that we have this type of response that very well fits the experimental one. And there are different parameters, but what is important is that we have two main tensorial components, and what is important is the ratio of these two ones that is given by the ratio of the two minima. So what we do is not fitting, is using a fast Fourier transform, and in every pixel we have these three information. So the mean second harmonic signal, that has not really a structure here, because it's in cornea, so in cornea the fibers are very small, practically it's homogeneous at the optical scale. But if we look at the phase of the different Fourier components, we can retrieve the orientation that is coded using this color wheel, so it's HSV coding, and you see that here we have fibers this direction, here this one, so we see the direction of the fibers in the collagen lamellae in cornea, while it's not possible with using another technique. So you see that there are also black pixels that corresponds to pixels where the quality of the signal is very low, because, for example, there is some cells here, so no collagen, and we have to filter out these pixels. So, just to mention, this is true only for cylindrical symmetry. So it means that it's valid for collagen. It's also valid for myosin, but it's not valid for tubulin. Tubulin is not cylindrical. And in plants, I think that it's not valid also for starch and cellulosis. So it has to be checked. So the last parameter was the ratio of the two main tensorial components of second harmonic signal. So this is a physical parameter. So it's not the relevant information for biological for biologists. So we have to understand what it means from the biological point of view. So here we practically it probes the organization of peptide bonds in the focal volume, so at some micrometer scale. So here we see that we have different peptide bonds that has a specific angle towards the main axis of the triple helix, so that's the pitch angle of collagen. And if we have collagen fibers well aligned within the focal, within the imaging plane, it's really, we can measure this angle, so it means that using this parameter, we can measure the molecular structure of collagen. But of course, it's the same in every tissue, except when there are some genetic disorders. So usually, if this parameter is not the usual one, usually it increases, and it means that we have some disorder within the focal volume. It may also increase because of out-of-plane orientation. So one way to use this parameter is not really in biological tissue. So still it's biological tissue. It's a parchment. So parchment is made of skin, animal skin that is stretched. So the fibers are perfectly in the plane of the parchment. And when we have some alteration of this parchment, the collagen is completely denatured. So there is no second harmonic anymore. But at the very, very first steps, there is some small disorder introduced within the focal volume before denaturation, and this can be measured using this parameter. So it's a very early stage of degradation. Here the degradation is higher in this direction when the denaturation of collagen is smaller. So this was done by my colleague Gaël Latour and Lauriane Robinet, who was attending Mifobio this year. Of course, we may have much more complex approaches of this polarization resolved experiment when we have forward analysis. So that's what's done by different groups, group of Barza, the group of Sophie Bracelet. But it's not applicable then in sick tissues. And there is similar approaches in myosin, and the myosin has not exactly the same parameter here, raw parameter as collagen, so it can be a way also to differentiate myosin and collagen. So just a take-home message for these polarimetric measurements. So it gives in-plane orientation and disorder parameter. So disorder parameter is either at pixel size, so the sub-micrometer disorder, or also in the field of view, if we compute, for example, the circular variance of the entropy of the angular distribution. So here is an example. Here we have a histological section of LC and keratotonic cornea. So it's transverse sections here. With LC, we see that all the lamella are well aligned. When it's not LC, there is some disorder, and accordingly, we saw some differences in the entropy. We can also do some mapping in 3D volumes. So here it's intact cornea. So the depth here is 600 micrometer depth, and we can do some polarimetric imaging up to 600 micrometer. The question is it valid or not? Because I have said to you, there may be some possible artifacts. So we have really to design validation protocols to check if it's valid or not. And here you may have some doubts because it's only always the same color. And practically for cornea, for example, as there is no ground truth about the structure of the collagen lamellae in depth, we have imaged the cornea in one direction, then rotated by 90-degree angle, and checked that the result with polarimetric images were also rotated by 90-degree angle. Also, to compare to other techniques, well, here, when we look just at the gray level image, we see some fibers. So we could do some image processing looking at the fiber direction this way. So if it's possible, of course, it's the best solution because it's much faster and less complex. But the only problem is that sometimes it's not possible in dense tissue. So, for example, you saw the images of the cornea. And also, it's not really at pixel resolution because we need different aligned pixels to see the direction. So, it's, of course, it depends on the application. If it's possible, do some image processing. If not possible, polarimetric second harmonic is better. Okay, so I've how many minutes? 5 minutes? Well, just to very rapidly comment on the third harmonic signal. So the specialist in third harmonic microscopy in my lab is Emmanuel Beaurepaire. So it's mainly images from his publications. So third harmonic generation is obtained in any protein, but in isotropic tissue, homogeneous isotropic, there is no signal. The reason is that it's very sensitive to the phase. And when we focus a beam, there is a shift of the phase of the electric field that is from pi, from minus infinite to plus infinite, but about 90 degrees in the focal volume. And due to the difference of these phases between the third harmonic beam and the excitation beam, there is a pi phase shift between here and here. So it means that it's destructive interferences. So to address this issue, there are two possibilities. The first one is to use bireferential materials so that we do not have the same polarization for the third harmonic beam and the excitation beam, or just to have heterogeneities so that we build the signal here, but here there is no third harmonic signal, a very small one. I mean, the amplitude of the third harmonic signal is different from tissues. So it means that we need micrometer scale optical heterogeneities. So here are some calculations for thin slab or thin beads, and you see that it's sensitive typically to diameter or thickness of 1 micrometer. And it was verified in epithelium. We see the cell borders, or here in lipid droplets. Here you can see a larger bead, it's a polystyrene bead, so that there is some signal at the edges, but not at the center because it's a larger bead, so at this place it's isotropic. Okay, and then I have some, I try to have yes, it works. So it's a very famous movie from Emmanuel and Nicolas Olivier about zebrafish embryo development. So it's really morphological imaging. So it's very efficient to look at this embryonic development. So we can switch to the next one. So we can also add some polarization. And just to briefly, it's similar to second harmonic. So it gives mainly a local direction and also some disorder parameter. So when we look at lipids, what it probes is mainly the main direction of the lipid bands. So here it's skin with here lipids from the stratum corneans, and you see these lipids, and you have also some structural disorder. Can be done also, here it's, yes. It's a movie of small biominerals within the ear of a developing embryo. And here you see that we have switched to fast third harmonic generation using electro-optical modulators, so it's possible to do it dynamically. And we can differentiate these minerals from, let's say, beads that are not crystalline beads using the modulation of the signal. More precisely, looking at this analysis of the signal. In every pixel, we can do some parametric analysis and then look if it's more single peak or more two peaks, double peak signal. It's a very basic analysis, but practically we have many, many parameters, and it's also very sensitive to optical aberrations, as given by this FTTT calculation by my colleague Nicolas Olivier. So that we can say that if in single peak, it's mainly vertical interfaces between these optical aberrations or horizontal interfaces, so it's signal from the surface. When it's double peaks, mainly birefringent bulk, and that can enable to differentiate different minerals. So it's kidney stocks from different pathologies. So you have monohydrate calcium oxalate, dihydrate calcium oxalate, or carbonyl hepatate, and we can really see that sometimes there is some signal from the bulk, sometimes from the interface, and here from the bulk, and here it's horizontal interfaces, and here it's vertical interfaces in this spherulites. So to conclude, we have different signals, so fluorescence, that's more basic, and these current signals that are more complex, and they have to be used in parallel because they are, well, these signals are free when we look at fluorescence. We just have to have another detector with another spectrum. Just to acknowledge all the advanced microscopy and tissue physiology group at Laboratory for Optic and Biosciences. So you maybe know many of the persons here. Here at Mifobio, you may see Pierre Mao and Chiara Strigarim, and also a few PhD students and postdocs. And just to mention also that we are recruiting research engineer in bioimage informatics through CNRS FSP. Thank you for your attention.
Thank you for your talk, very interesting, very didactic also, helpful for us. I was wondering if it's possible to do some second harmonic generation, or maybe the others also, on non-traditional in vitro models like organoids or organ-on-chip systems. Do you know if it's compatible with thick 3D constructs that are not...
Yes, when there is some...
plastics in between, or gels, or...
Yes, but usually there are also collagen fibers within these organoids, so they can be visualized using second harmonic generation. Yes, it works in any signal. Due to the origin of the signal, it works in fresh sample, in vivo, ex vivo, fixed, frozen, in any signal. For third harmonic generation, it may be sensitive to the different layers, so sometimes there may be too much background noise if there is a gel or something like this. But otherwise, I think it's okay.
Thank you.
Other questions? For myself, I have a naive one. I missed why you said that you cannot detect tubulin with the polarimetric SHG.
We
can do it. We can do it with polarimetric, but usually the interpretation of the images of the polarimetric response is to say we see a pitch angle of an helix, but there is no helix in tubulin. That's what I say. So this interpretation is not the right one. So my colleague Chiara Stringari has done some calculation, and the paper is under review, so it will be published very soon, I think so.
Thank you. Thank you very much, Marie-Claire, for this very deductive talk. I was actually wondering, since we are at Module 5, but in parallel to Module 6, actually, there are common questions that we have. I was wondering whether you can stress a little bit on how the variation of the SHG signal can inform us about the mechanical properties in time of the tissues related to the young modulus, for example, and whether it is possible to extract more quantitative information from that. And
Yes, it's possible. Actually, I've not presented this results, but we have done some combination of mechanical. Well, we have done some mechanical assays under the microscope. So to really elongate some skin and the tendon, and also we are doing it with the cornea, but here it's just other types of assays. And we are looking at the deformation, while with the mechanical assay we can measure the force, and then we have a young modulus. So it's possible to do that. And also we see also that the anisotropy parameters, the ratio of the two main tensorial components, give an idea of the alignment of the fibers within the focal volume. So it also can be used in this type of mechanical measures. So that was done with my colleague Jean-Marc Alain at École Polytechnique.